Hello, welcome back. We are getting ready to go into meiosis, which is very similar to mitosis, but some very important differences that I'm going to try to point out to you. And then at the end of this lecture, I will do some comparisons between mitosis and meiosis to make sure you're clear as to the differences. Before we jump into some of the textbooky type of stuff, I wanted to try to make some things very clear because I know a lot of times meiosis, people are pretty okay with mitosis. Meiosis is where things get a little bit confusing. So I wanted to first off talk about kind of the chromosomes that we're going to be seeing, some of the terminology that we're going to be seeing, because if you can keep track of your chromosomes and keep track of your terminology, meiosis will go a lot easier. So first of all, when we are talking about our chromosomes, it's more than just replicating them and pulling them apart like we saw in mitosis. Some really special things are going to be happening to our chromosomes. So I wanted to make sure that everybody's comfortable with the idea that what a chromosome is, right? So a chromosome is just a piece of DNA with your A's, T's, C's, and G's, right? So your chromosome is one big long piece of DNA. When that chromosome goes to the S phase of interphase, what happens is, this is what we're going to learn the details of in chapter 12, but what happens is that DNA actually splits into two identical copies. So here, one half splits, the other half goes over here, and then the new strand, so I'm going to write that in a different color. So each half of the original DNA becomes a template for the daughter half. And so these are going to have the same exact sequences of A's, T's, C's, and G's. And they're held together by this dense area here called the centromere. Okay. So when we have just a single chromosome, sometimes we call this a single chromosome, or I might call it an individual chromosome or unreplicated chromosome. Okay, it's all by itself. This would be what the chromosomes look like in G1 and after the end of anaphase, if we're going to try to find a spot for them. These are called duplicated or sometimes called replicated chromosomes, because as you can see, they are replicated, but because they're held together by the centromere, they're still considered one chromosome. If this is chromosome number 1, when it's an individual chromosome, this is still just chromosome number 1. It's one chromosome. We haven't added any new information. We just have a copy of it. And these copies, each half is called a sister chromatid. You saw that word in the mitosis, but this is going to become really important when we talk about the meiosis, knowing the difference between the centromere, the sister chromatid, a replicated chromosome versus an unreplicated chromosome. So this is what's happening during the S phase of interphase, is all of the chromosomes go from this individual form to the replicated form made up of two sister chromatids, which are identical to each other. They are identical, okay? So that's one piece I wanted to share with you. So this is gonna be fun, erasing all these bits. So that is some of the terminology for the difference between a replicated chromosome, an unreplicated chromosome, and the chromatids and the centromere and all that kind of stuff. So there are all these little spots. Okay. So now what we need to do is understand the difference between these replicated chromosomes and homologous pairs. Okay, so I'm going to try to use some color here. I'm not going to do the different color for the replicated. I'm just going to do kind of some solid colors. So let's say I have chromosome number 1 from mom, and I have chromosome number 1 from dad. Okay, so these are both chromosome number 1. They have the same genes. They might be different forms of the genes, which we'll identify that in a little bit. So let's say mom's got a gene up here on her sister chromatids, because they're exactly the same. It's going to be the same gene for, let's say, taco tongue. This is the gene for taco tongue. You guys know what taco tongue is, right? Taco tongue. And let's say dad's got the same gene over here, but he's got taco tongue where he can't do it. So dad's tongue can't taco, but mom's tongue can. So it's the gene for tongue musculature. So it's the same chromosome, chromosome number 1 from mom, chromosome number 1 from dad. They both have the gene for tongue musculature, but they just have different versions of it. Mom can do taco tongue, dad can't. These chromosomes are called homologous pairs. They're partners. They are the same chromosome, chromosome number 1. If I wanted to do other chromosomes, I could do maybe chromosome 15, one from mom, one from dad. These are homologous pairs. I could do chromosome number 22, right? One from mom, one from dad. They're the partners. They're going to have the same genetic information, albeit maybe different versions of it. So maybe eye color, hair color, height, taco tongue, knuckle hair, whatever it happens to be. These are homologous pairs. So homologous pair, homologous pair, homologous pair. And all of these, how I've drawn them, are in the replicated form. You could have, if I wanted to draw them in the unreplicated form, I'll draw it over here. So this would be unreplicated form of chromosome number 2. They're still homologous, they just haven't gone through the S phase of interphase yet. Okay, so hopefully this is helping you kind of understand our chromosomes. One last thing before we move on is, remember that centromere that holds those replicated chromosomes together, right? Even individual chromosomes have centromeres. So you're going to have to learn how to count chromosomes in trying to figure out whether they are duplicated or not. Not duplicated or not, but if you have one copy or two. So I suggest counting centromeres because this has 2 chromosomes. I have 2 chromosomes, 2 chromosomes, 2 chromosomes. Don't let the chromatids make you think that there's 4, right? You might think that that's 4. It is not 4 chromosomes. It's 2 chromosomes that have been replicated. They are 2 replicated chromosomes. And I know that because there's 1, 2 centromeres. These are 2 chromosomes. That's really easy because there's 1, 2 chromosomes, or 2 centromeres. So keep that in mind. Count the centromeres. The replicated chromosomes count as 1 chromosome. Okay, you ready to move on? Let's get to it. Okay, so here I have a general overview and the purpose of meiosis. So when we were doing mitosis, mitosis was cells dividing to make more identical copies for growth from like a fertilized egg to a multi-trillion cell organism, for repair, like if you scratch or damage yourself, or maintenance, like you have to continue, producing new skin cells to cover your skin. So those are the major functions of mitosis. For meiosis, the only goal for mitosis is to make gametes. Excuse me. And what are gametes? They are your sex cells. They are your sperm and egg. The only time you do meiosis is to produce gametes. And location, where are these things produced? Sperm are produced in testicles, eggs are produced in ovaries, and that's it. Everywhere else in your body is going to go through mitosis when they do cell division. It is only in your gonads, the eggs and the gonads, ovaries and testes that produce the gametes. So gonads for gametes, right? It's like a slogan, gonads for gametes, or gametes from gonads, however you want to think about it. So that is the sole purpose of meiosis, is to generate sex cells, eggs and sperm. Now, when we talk about our genome, if you remember, we talked about the genome at the beginning, we have 46 chromosomes. We have 23 pairs. And if you remember, I started at the beginning, you have one from mom and one from dad. Well, how did you get that? Well, they came separately and they came during fertilization, this event that's happening right here. Shortly in that picture will be fertilization. Dad brings in one half of the chromosomes, his 23, and mom has her 23. So again, the goal of meiosis is to make those gametes that have half the number of chromosomes. So somehow we have to go from stem cells that have 46 chromosomes to sex cells that have 23 chromosomes. And the process of meiosis allows us to do that. So here is just this overview. We're going to take a look at this process. So in this particular example, we have 4 chromosomes, right? We can count those centromeres. 1, 2, 3, 4. We have 4 chromosomes. That's our diploid number. Okay, then they're going to go through replication. That's going to be S phase of interphase of meiosis. And now we are going to count our centromeres again. We have 1, 2, 3, 4. We still have 4 chromosomes, but they're replicated chromosomes. So these were individual and these are replicated, or they're saying duplicated, right? Unduplicated and duplicated. So now they're replicated. We still only have 4 chromosomes. Then it goes through the first round of meiosis. There's 2 stages. There's meiosis 1 and meiosis 2. At the end of meiosis 1, how many chromosomes do we have in each of the daughter cells? We'll count our centromeres. We have 1, 2. We have 1, 2. So we just went from 4 chromosomes at the beginning of meiosis 1 to 2 chromosomes at the end of meiosis 1. So we have cut our number of chromosomes in half. We've gone from what we call diploid to haploid. And now we're not done. So even though we've gone from 46 to 23, our chromosomes are still in the replicated form, right? So they're still in the replicated form. We cannot make eggs and sperm with replicated chromosomes. So now we have to go through meiosis 2 to split those replicated chromosomes into individual chromosomes. So let's count our centromeres. 1, 2, 3, 1, 2, 1, 2, 1, 2, 1, 2. So now we have daughter cells that have half the number of chromosomes, right? Because we started off with 4 individual chromosomes. We had 4 duplicated chromosomes. Then we had 2 duplicated chromosomes. And now we have 2 individual chromosomes. So we start off with 1 parent cell, we get 4 daughter cells with half the number of chromosomes, and now these will become our egg and sperm. So that's the overview of meiosis. Now we're going to jump into the details of each of the steps. Oh, I forgot. We're going to talk a little bit more about genes and alleles. So kind of like what I did at the beginning of the picture is this could be our mom, and this could be the dad. These are homologous chromosomes. They have the gene for flower color, right? Flower color. Mom happens to have a purple version. Dad happens to have a white version. It's the same gene, flower color, but a different version, purple versus white. So these varieties of genes is this word that we call allele. We're going to see that a lot more in chapter 11 when we get to the genetics. So genes code for a particular trait, and alleles are different versions of that trait, right? Taco tongue or no taco tongue, purple flowers or white flowers, tall plants or short plants, 6 fingers or 5 fingers. Those are all the genes, and the different versions of those genes is what we call alleles. Right? And then here's just another slide showing you the homologous pairs. I forgot I had this in here. I did the drawing at the beginning, but it's okay to see it again. So if I'm going to draw my centromere, right? We have 2 individual chromosomes, a mom and a dad, They're a homologous pair because it's the same chromosome, 1, 1, 2, 2, 5, 5, 15, 15, whatever it happens to be. They would have the same genes, maybe different alleles, but the same instructions for whatever the character is or the trait is. And then when they go through replication in S phase, tried to bring in our knowledge from prior learning. And so now we have a homologous pair of replicated chromosomes. And again, those pieces are the sister chromatids. Okay. Now here we can define our words. I've mentioned these a couple times already, but ploidy stands for how many sets of chromosomes you have. We have haploid is 1 set of chromosomes. Diploid is 2 sets. Okay, so this is where you're going to see your homologous pairs. And these are just individual. Well, these are individual as well, so maybe that's not what I'm trying to say. So these are, you just have one number 1, one number 2, one number 3. That's your haploid. Diploid is you have 2 number 1s, 2 number 2s, 2 number 3s, so forth and so on. Okay, now we get to it. So similarities of mitosis, meiosis. 1, they go through the same steps. They both have prophase, metaphase, anaphase, telophase in that order. They just do it twice. So there's 2 steps, but different things are happening. So interphase, so this is going to be your G1, S, we're going to replicate all of our DNA, G2, we're growing, all of that's the same interphase between mitosis and meiosis. In prophase 1, we have all of those same events that we saw in mitosis. We have the degrading of the nuclear envelope, the nucleolus disappears, the spindle starts to form, the centrioles spread to opposite sides of the cell, the chromosomes start to condense. However, a very important event happens here in prophase 1. This is what we call crossing over. I have a slide that's going to show that. But what happens is the homologous chromosomes, right? So I don't have enough fingers. But if this was a sister chromatid of a mom and this was a sister chromatid of the dad, they swap over and they actually break and rearrange their parts. So you can kind of see that a little bit in this area right here, right there, where the blue and the red parts swapped pieces. So this is what's called crossing over, and this delivers or it allows for genetic diversity in sexually reproducing organisms. So the homologous chromosomes come together, they partner up, they're replicated, so they've got their 2 sister chromatids, they partner up, they do some crossing over, that's prophase 1. And then in metaphase 1 is another really important event for diversity. This is where we see what's called independent assortment. Independent assortment is where your homologous chromosomes line up on the metaphase plate, not individually. It's not going to be chromosomes lining up like this with the spindle, like what we saw in mitosis, where all 46 would be lining up on the metaphase plate. In, I'll do a different color. In metaphase 1, the homologs line up like this. So the partners are lining up because what's going to happen in anaphase 1 is those are going to be pulled apart. And that's what we see here in anaphase 1. The homologs get separated. They're still replicated though. So again, if we're wanting to count our centromeres, by the time we get done, we start off with 4 chromosomes, right? 1, 2, 3, 4 replicated chromosomes. By the time we get done, we have 1, 2 in that cell, and 1, 2 in that cell. So we've reduced our number of chromosomes by half, but they're still replicated. And now you can see a little bit clearer that crossing over event where they swap pieces like what we can see down here in that example. So that's the end of meiosis 1. We have 2 cells with half the number of chromosomes. They're still in their replicated form and crossing over has taken place. So here's a little bit more of a close-up of what happens in that prophase 1, where the homologous chromosomes come together. They actually physically break bonds of the DNA. They swap pieces and then reattach. So you end up with these recombinant chromosomes that if those are the eggs or the sperm that get fertilized, you're going to have a new combination of genes. It's going to be mostly mom with a little bit of dad, or mostly dad with a little bit of mom. And this recombination event is completely random. So sometimes it happens a lot, sometimes it happens a little. So there's no way to determine when recombinations are. We know it always happens during prophase, but on what chromosome, it's completely random. You might get a lot, you might get a little. Okay, so this that independent assortment that happens in metaphase 1 is, again, we have our homologs coming up on the metaphase plate. They could either, I know this, we're only showing 4 chromosomes here, but let's say all mom ends up on one side and all dad ends up on the other side. That's random, because the other possibility is half of mom on one side and half of dad on the other side. And if we have 23 pairs, right, chromosomes 1, they could be on either side of that metaphase plate, which is going to give you various combinations of genes. Now, I know they're just showing 2. It would be really hard to show all 23 pairs, but that's the idea of independent assortment and crossing over combined to give you huge amounts of genetic diversity in sexually reproducing organisms, which is a benefit because it gives us the ability to fight off diseases and pass on traits that are beneficial for the environment that we are in. Okay, picking back up. So at the end of meiosis 1, we had 2 cells with half the number of chromosomes, but the chromosomes were still replicated. And so that's what we start off with here. We have our 2 daughter cells of meiosis 1. They go through prophase 2. So they kind of unwound and put the nucleus back at the end of telophase 1. And now we got to break it all down again. Our spindle forms. Metaphase 2, we have our replicated chromosomes lining up on the metaphase plate. Anaphase 2, we're pulling apart the sister chromatids. This is very much like anaphase in mitosis. And then telophase 2, everything kind of comes back around. We get our nuclear envelope, the chromosomes uncoil, the spindle breaks down, nucleolus reappears, and what we end up with is 4 haploid, right? So half the number of chromosomes cells, which are going to be your sex cells. So if this was a male, these would be sperm. If this was a female, they would be eggs or plants, or what would have spores, pollen, and eggs, and those types of things in the plant world. So this is our ultimate goal of meiosis 2, or meiosis in general, but specifically for meiosis 2 is to end up with these 4 haploid daughter cells. And this last slide here is just, again, comparing the processes and the outcomes of mitosis to meiosis. So we start off with, they both are going to start off with a diploid cell with a chromosome number of 6. They're going to replicate. So again, this is S phase of interphase for both of those. In prophase of mitosis, you don't get any crossing over. They're just kind of hang out, getting hooked onto the spindle fibers. In prophase 1 of meiosis, this is where you get the pairing up, and this is where you get the crossing over, right, in prophase 1. In metaphase of mitosis, every single chromosome lines up on the metaphase plate and attaches to the spindle. In metaphase 1 of meiosis, your homologs line up like this. So the partners line up, the replicated partners line up. In anaphase of mitosis, the individual replicated chromosomes come apart. And if you take a look, we started with 6, we end with 6. In meiosis, we do the metaphase 2, all the replicated chromosomes line up. We split. Here we have 3 chromosomes. We started with 6 and we end up with 3. We have 4 cells that have half the number of chromosomes. And if you'll notice, there's a different genetic combination due to the crossing over and the independent assortment. All right. Whew, that's a long talk about meiosis and cell division. So for both of these phases or processes, mitosis and meiosis, please make sure that you can go through and outline. You can describe them in words if you wanted to. Be able to maybe draw them was a really good way to practice what happens in each of the stages of prophase, metaphase, anaphase, telophase for mitosis, prophase, metaphase, anaphase, telophase 1 and 2 of meiosis, making sure, because it's really easy to get them mixed up and jumbled around in your brain. So the best way to do that is to draw it out, to write it out and talk through it and make sure that you can go through it on your own without looking at anything. And that's going to let you know whether you know it or not. Okay, that's the end for meiosis. I will see you next time. Bye.